problem with almost all of these scripts is that they're very difficult to benchmark, because there's not really always a right answer from a metagenome project. So just because you use this pipeline or that pipeline, it's not really too easy to say which analysis is correct unless you have a wealth of biological data, or you have like a replicate somewhere, like a completely replicate environment somewhere that you can compare it against. And most of these scripts have a high number of dependencies, particularly any kind of script that includes annotation. So this can cause problems when you start to require the dependencies or the databases that have to come with these wrappers. That it can take up a lot of space. And if you're trying to use your computer cluster, if you don't want to use the wrapper that they have already downloaded the dependencies for, a lot of times you're just out of luck. And so if your use case doesn't fit into the use case that the wrapper was made for, then you kind of end up having to do everything yourself step by step. So this is why in our group we've made our own sort of wrapper scripts, and that way we can write them for our use cases, and we've got the dependencies and the databases that we need on our local machines or on the cluster. And so basically for this, I've separated the different steps into these kind of 5 different scripts, that some of them are just for shotgun sequencing analyses, and some of them are a little bit more flexible, like quality control, that we can just use for any kind of sequencing project. So we separated into these 5 steps where we do quality control, taxonomy assignment, assembly, binning, and then annotation. And they're not necessarily a linear use case, as we'll see in a moment. Okay, so the first thing though is that we usually want to do quality control of the reads. This is a standard process, so that there's debate about whether or not we need to do it, especially for current sequencing technologies like HiSeq, where you have just hundreds of thousands to millions of reads for a sample. There is an argument to be made that quality control is not entirely necessary, but we use BVDoc as to remove any residual adapters, because adapter sequences that are present on any of the reads can really impact the assembly process. And then we try to get rid of any reads that have too low quality of base. So we first trim those down, and then any reads that are below a certain size threshold, after quality trimming, we discard. And our pipeline also includes a function for CLR reads, because we've found that, like a lot of people, that hybrid sequencing of metagenomes can really improve the reliability of your results. So we work that into our analytical pipeline as well, but it's not a requirement. So after BVDoc, we usually want to output some kind of metrics, right? So you can see what's happened. So we use FastQC, and then I use MultiQC to aggregate FastQC results, which is not super necessary when you're doing a shotgun project, because you might only have like 3 or 4 different samples. But when you're doing marker gene sequencing, this MultiQC aggregator can really help to view FastQC results. So that's why I like it. So for the rest of this presentation, when I'm talking about the scripts that we use in our lab, I've done just a quick analysis on one of our old metagenome samples, which is called KAS, which is a highly enriched microbial consortium that showed some Animox activity, and it was sequenced with NovaSeq 6000 2x150. And then I just went to the SRA on GenBank and pulled down some random project, SRR2394994. And the project title was Analysis of Biotic and Abiotic Drivers of Community Assembly. It was from Brazil, I think, this project. But I just pulled this one down as some kind of example data. And it was a HiSeq project with 2x250. Okay, so throughout, I'll just kind of use these 2 sequence read sets to demonstrate what we get out of our scripts. Okay. So here's an example output from the readsQC script. So this on the right-hand side, this is just images of what the MultiQC produces after you run it. The MultiQC report itself is quite clickable. It's very nice. Strongly recommend if you don't use this program. It's a pretty good one. But I can't quite figure out how to get the links to embed in PowerPoint reasonably. So I just had to take a picture. But you can see that it will give you some nice summary of the per sequence GC content. So you can see if there's any outliers or if you have like something very strange, like two distinct peaks, which will tell you that there's something wrong with your reads. It counts duplication events for you. Yep. So you can see if you have too high level of duplication. So that might indicate that your sequencing depth is extreme. So you might need to cut down your read set or something like that. And it can give you a quick highlight of anything that looks abnormal. Sometimes it looks abnormal because of the type of sequencing that you've done or because of your sample. Like if you're using an enrichment instead of a random environment, instead of just an environmental sample, a lot of times it will show you it will show you an error, even though there's not one. So it's just a good heads up to see if there's something wrong. And then it gives you a Fred score analysis along the length of the read. So you can see like, maybe I need to go back and cut my reads because I'm seeing some sort of degradation in the read quality at a certain length, something like that. And then BBDuck, of course, puts out some kind of metrics to show you if there's some real problem with your reads, because most reads these days come with the adapters already removed, and depending on where you get them done, they should already be quality trimmed. We don't use BGI anymore because it takes too long to send the samples to Beijing. But the BGI used to always do the quality trimming for us, and now we use Novogene in Singapore, and they do, but they still miss some. So this is why it's important to run this through your own program. So here, for example, you can see that the SRR, the random one, we lost about 22% of the reads, something like that, and most of that was. Oh, sorry, I cannot read your questions. Oh, hi KK. Let's see. I'm sorry, BJAA012. I cannot read your question. So maybe somebody can read it to me after. So yeah, and so this KAS, this one, you can see when we put it through, it's got quite a lot more reads. But this is why I like BBDuck, because it scales very, very well. So this has about 6 million reads and take about 10 minutes to run. And this one has quite a lot more, right? Like 5 times, 10 times the number of reads, something like that. And it takes just about twice as long to run. So BBDuck is pretty quick. That's why I like it. So after quality filtering of the reads, this is when your experimental design should tell you what to do next. So when we talk about metagenome assembled genomes and things like this, if you're actually looking for getting a genome out of your assembly, you really want to assemble the reads, because otherwise you'll never get a genome out of it. If you don't care about getting a specific genome out of your sample, then there's no need to assemble the reads. Actually, you can just go directly to annotation and taxonomic characterization of the reads. So this is why it's important to have this sort of plan before you start, because you can end up wasting a lot of your time doing a computation that's just not necessary. So this is also why in our group that we separate the functions into different scripts so that you can select which things you actually need to do. So sometimes though, the first thing you want to do no matter what is to investigate the phylogenetic diversity of your reads. And so for that, we have this reads taxonomy script, and our reads taxonomy script uses two different tools to assign taxonomy to the cleaned reads. So we use Kraken2 and Bracken and Metaflan 4. So we use these two because they have a different approach to taxonomic classification of the reads. Kraken2 Bracken matches k-mers to a pre-built database. So this tool will break your reads apart into defined k-mers of a defined length and match them against an index database that you can access online. You can download it. And then it will count how many reads will match to each of the segment in that database and return a result. Metaflan 4 takes a different approach. Metaflan 4 takes merged reads and tries to match them against an indexed database of marker genes that it associates with different phylogenies. So we like to do this twice because the results differ a lot. So for example, here are the two analyses, one from Kraken2 Bracken and one from Metaflan 4 for these two read sets. So you can see that this SRR, both of the methods find that it's dominated by this methanobacterium, but the degree of dominance differs. And we have a pretty high level of this methanosarcina also in both analyses. And we have some level of this methanothrix in both. But you can see that the proportions differ quite a bit, and then the minor populations differ pretty significantly. So this is what I mean when I say it's a little bit difficult to know which one is correct and which one is incorrect. So if you really wanted to answer this question about the structure of your community, it's usually a pretty good idea to include marker gene sequencing, because 16S RNA gene sequencing, for example, will give you a much higher resolution of the community. So a lot of studies that are focused on composition of an environmental sample will include marker gene sequencing along with their shotgun sequencing, and this is why. So similarly for KAS, which is a highly enriched culture, you can see that there are similarities. So we find this Candidatus quinina in both of them at a very high fraction. But in one of them, we see a minor population of Acidovorax and then metaphan-4. And in Kraken, we see a minor populations of Diaphorobacter and Eusorhizobium. So this is why we use both of these. And then we kind of have to use later analyses to determine which one of them we think is more correct, or which one of them is more meaningful for the type of analysis that we're going to do. Okay, so read assembly is almost always what people are hoping to do out of their shotgun metagenome projects. So we have a dedicated script to perform this task. And just like with taxonomy assignment, we take a multiple tool approach. So we use Metaspades and MegaHit and IBDAUD. And for projects that include CLR reads, we also run Fly, just because Fly requires CLR reads. Otherwise, you would use all four. So a lot of wrapper scripts will pick one of these, but it's generally a good idea for your own project to use at least two, because much like the taxonomic classification of the reads, the assembly of the reads can differ quite significantly between the different methods. So even though all of these things are De Bruijn and graph assemblers, they will identify different contigs and different scaffolds. For all of these, we use Pylon to polish the contigs after assembly, and then we do a contig comparison with Quost. So this is a pretty standard approach, except that for us, we like to break this step out. So a lot of wrapper scripts will bury the assembly with the binning. But I find that if you do that, you don't get the chance to evaluate which tool actually is best for this particular set of reads. So here's an example of a Quost output of an assembly that we did for the KAS. Right? So you can see that the different tools will actually give you quite different results. The polishing has sometimes large and sometimes minor effects. So in this read set, the polishing was not super productive. But you can see that the different tools give quite different numbers of contigs. So here you can see we have a little bit more than 3,400 that are longer than 1,000, or just under 4,000, or a little bit more than 3,000. So here it's in MegaHit gave me the most contigs, and so you might be inclined to think that's the best assembly. But when you look down at the length of the longer contigs, or the total number of bases in the assembly, here Spades has done a better job. But if you look at overall coverage of the high coverage reasons, right, you can see IBDAUD has done the best job. So there's all these different metrics that you can use to evaluate an assembly. And this, again, will depend on what you're going for, right? If you're really curious about a single genome, you can just select this longest contig and have a look at it and see if that's the genome that you're interested in. And then you might want to pick the assembler that has done the best job on that contig. Or if you're most curious about getting the greatest representation of possible things that are in there, you might focus on the assembly that gives you the largest number of total bases, something like that, right? So here you can see that Spades has the worst high coverage, even though it's got the most bases. Yeah. So for more enriched genomes, this is one advantage also of giving yourself a chance to look at the intermediate files. Is you can look at the graphs. So you can load the graphs into something like Bandage. This is just a Bandage plot. And a lot of times, if you have a pretty enriched metagenome, you can find a path through the contigs at this point, or you can find multiple paths through multiple contigs and give you some idea of how many genomes and how complete those genomes are going to be by the time you get to assembly at the very end, like reassembly, sorry. Okay, so this is why I generally say that assembly should be chosen based on your downstream needs, not based on specific metrics. It's very easy to write a code to pick the assembly with the largest N50, but it's usually not a great idea, because if you pick the biggest N75, so this is the metrics from the SRR project. You can see that here the IBDA has by far the biggest N75 and L75, but it has far fewer intermediate sized contigs than the Spades assembly, and also has a much shorter longest contig than the Spades assembly. So it's a little bit more work to do it this way, but we find that it gives us better results. Okay, so after assembly, you can go directly to annotating the contigs if you don't care about the genomes themselves, like putting things into bins and finding which contig belongs to which kind of taxa. You can then pass these things directly to annotation. You can actually annotate with Eggnog. So we use Eggnog Mapper in our group for this, because it runs pretty fast locally and it's nice and flexible. And you can run Eggnog using just reads, but it tends to do a better job with contigs, because it does a better job when it can call its own proteins. So, but you can do it without. It just the results are not always great. So Eggnog Mapper, we like it because the database is comparatively small. It's only about a 40 gigabyte database compared to some of them, which are hundreds of gigabytes, and it can run Diamond, HMMR, and MMSeq alignments for different annotations. And so again, we take the same approach that we take with assemblies. We run several different annotations, and then we can compare the outputs of those different annotations, because annotations are inherently inexact. And so we like to use different alignment methods, like MMSeq and Diamond, and then also I like to align it against different databases. So we like to align it against a cake database and a COP database and a NUC database or whatever. And you can get quite different results. So here's just an example of COG assignment of amino acid sequences from the SRR project. So you can see that this is just it's pretty standard output of the annotations. Sorry, did I go the wrong way? Yeah. Okay, so after annotation or before annotation, if you're actually caring about binning of the contigs, we have our binning script. And much like annotation and taxonomy, for binning, we take a multiple approach. So we have this binning script that uses one or more of the assemblies that you can select, and then it calculates the abundance of the contigs and the CDS, maps quality filtered reads, and then we use 3 different binning tools. We use Concoct, MaxBin2, and MetaBat tool. And then we take the outputs of those and feed them to DAS tool. And DAS tool does a bin rectification and evaluation. And it selects what it terms high quality bins and passes those to CheckM. CheckM is a standard pipeline for assessing bin metrics. And then our script just puts out some tab separated files that we can use for evaluation. But it does take a long time, right? Especially when you've got multiple samples or multiple metagenomes and you decided that you wanted to use one or more of the assemblies to compare. This is when at this step, I guess, is really when metagenomics sequencing projects start to take up a lot of time and space on your computer. It's because for each of these methods, you produce a large number of output files, and as you start to map things to multiple assemblies or bin things multiple times, the project can start to take up a few hundred gigabytes of space, at least temporarily, on your hard drive. So this is one of the reasons that if you're able to run these things on your cluster, it's a good idea. But getting all of these dependencies onto your cluster can be difficult. So we're generally not able to do it on our cluster at NUS. Okay, so this is binning. And so this whole process can take a very long time. So that's why if you don't care about the bins, it's not a good idea to run this, which is why we separate it from the rest. So this is just an example from an old project about the types of things that come out of DAS tool. So this is what I mean when I say the number of results starts to explode. So if you have multiple assemblies, DAS tool wants to produce optimized bins for each of those assemblies, and then checkm will want to analyze each of those sets of optimized bins. So if you choose 3 of the assemblies, you're going to end up with 3 different sets of outputs. And each assembly is going to have some number of bins and some number of mapped files, and it just starts to take up a long time. But if you can automate things like the bin selection with DAS tool, you can save yourself some effort. So DAS tool uses what it calls unique single copy genes, divided by the number of referenced single copy genes that it thinks should be in each bin, and this is how it evaluates the quality of the bins, and it produces some kind of volcano plot for each of the bins that are in the input file. So here we see we got concoct, maxbin2, and metabab bins. This is from the KAS project. And so this is fairly typical of how the binners usually perform. Concoct usually has a higher number of bins, but it has a tendency to produce more lower quality bins. Metabat usually has the fewest number of bins, but they tend to be a little bit higher quality. And so Metabat then correspondingly usually has a higher number of unbinned sequences. And DAS tool will align all of these the contigs up from these various bins and make a composite set of bins that it deems to be high quality. And you can tune that threshold for high quality here. It must be set at, I guess, 0.8. Yeah. And then it will take those bins and tell you that those are the good ones. And you can pass those on to downstream analyses. So there are other bin rectification tools, but I found pretty good results with DAS tool. Okay, so those high quality bins, so-called high quality bins, are then we pass them to checkm for just a general kind of analysis. Checkm is a good tool, but it is a bit old, and so you have to be pretty careful when you use it. So checkm that uses a different kind of evaluation metric for the bins. It will map the reads back to the bins, and it will estimate abundance of the community based on the number of reads that map to each of the bins and the number of reads that map to the entire initial input data set. And it will try to assign taxonomy to the reads based on like SSU analysis, but it has its own SSU database that it uses to assign that taxonomy, and it also uses Busco to assign taxonomy. So a lot of times you get these kind of seemingly conflicting results. So for example, in this project, we came out with 8 high quality bins from DAS tool, and then of course the unbinned contigs. And this is not uncommon, where the unbinned contigs are a pretty major fraction of the entire data set. But a lot of times, if you look at them carefully, it's because these contigs are fragments. They may be like 500 or 600 base pairs long, but sometimes not. So you have to take care about the unbinned contigs. So, but in this case, you can see that if we compare the taxonomy assigned by checkm to the bins themselves, that it agrees a little bit with the reads taxonomy assigned by Kraken and Bracken or Metaflan. So for example, this bin here, this bin 5, is a 3.54 MBP genome that comprises 68-ish% of the total reads. And so it's like 68.71% of the binned community, 78% of the binned community and 68% of the total community. And the SSU place it as a planktomycetes, which is where the Anammox bacteria are, which is where the Quininia are. But you can see that the marker lineage that it's assigned is just bacteria. So it's a very general marker lineage. And this is something that we commonly see in checkm, and it's because the databases are quite old. And you can update them, but it requires you to fiddle with the base code in checkm, which is maybe a little bit difficult to do. So here you can see that it agrees more or less with the Kraken 2 and the Metaflan taxonomy assignment, in that this bin is the most abundant bin, but the numbers themselves are quite different. Okay, so similarly, we can see that there is this kind of mesorhizobium. So this is probably this one, this rhizobiales and this Phylobacteriaceae. This is 3.6%, this being 8. But you can see that we also have a rhizobiales up here, which is 0.38% of the community, and another one right here, which is 0.9% of the community. And each of these bins, the genome size is quite different. So it's possible that all 3 of these bins, 2, 4, and 8, represent the same genome. But it would take a little bit of work to figure that out. You'd have to go actually into the bins themselves, because the binners have separated these reads for a reason. So this is when you will want to go back to the DAS tool results, and you can see in DAS tool like where which contigs were in each bin originally for each binner. And you can also go back and look at the predicted amino acids and the nucleotide sequences and try to find out why those bins were put where they were put. And within this directory here, there's also the metrics, the DAS tool metrics, and CheckM puts out metrics as well for each of the bins. So you try to figure out why these bins were separated and if they're actually the same thing. Okay, so similarly, Metaflan found this acidovorax, which could very likely be this one up here, this Burkholderealis. It's about 8% of the community about 9% of the community in bin 1 here. So that's quite a lot more than this 3.66, but there's not really any way of knowing, because also similarly could be this one. So it's hard to know. So if you add those 2 together, that's quite a lot more than 3. Yep. But this Comamanidaceae could also be this Diaphorobacter. So it's hard to know which is which, really. So this brings us back to why it's important to do different, to take different approaches to things like taxonomy assignment and genome assembly when you're doing these projects, because you really need to feel your way through your data. And if you don't have biological data to back up any conclusions that you make, your conclusions lose a little bit of their support. Okay, so CheckM also does what it calls a quality analysis of the bins, and it uses the terms heterogeneity, contamination, and completeness. Heterogeneity means that the bin has multiple copies of things, that there should only be one copy of. Contamination means that it has something from another lineage that it shouldn't have, and completeness means how many things that it does have, right? So because CheckM uses Busco, right? So Busco uses these marker lineages, and these marker lineages are predefined in the Busco database, and it will assess the number of Buscos that are complete, the number of Buscos that are missing or duplicated, or the number of Buscos that don't belong to that lineage. And then there's a terms that are used to describe those. But the Busco database that CheckM uses is actually very old. It's from I think 2013 or 2015. So this Busco analysis from CheckM should always be replicated with the bin sequences of your interest with a more updated database. But you see this kind of figure in a lot of papers, and people don't always pay attention to that part of it. So after you've done that binning and you've selected your bin of interest, so for example, here we pick bin 5 because we're interested in this Anammox bacteria, right? So we think that it's probably this one, right? So we think that it's probably this one. It's a fairly complete bin, and it's not very heterogeneous, and it doesn't have any contamination. So we move ahead with annotation of that. Oh, sorry, there we go. So here's just an example of the annotation of that bin. So I've taken the KEGG annotation of the bin and then mapped it onto, use the KEGG mapper to identify the KO. So this is the central carbohydrate metabolism. So a lot of times when you're doing when you're trying to get a genome out of a metagenome project, this is the type of picture that you're kind of looking for. And so this would be the step at which you can do that, right? You can actually do a metabolic analysis of it. But you need to be careful when you do it, right? Because just because you've done an annotation doesn't mean that your annotation is going to find everything that you're looking for. So for example, we know that this enrichment culture had Anammox activity, but when we look at the KEGG annotation, we can see here that there's none of the genes that we know are necessary for the Anammox reaction, like the hydrazine synthase. But this could be due to a lot of reasons, like we talked about at the beginning, like that target gene has just been washed out somehow, either during the sequencing or during the analysis, or it could be that it just got missed in the annotation. That happens a lot, particularly for proteins that are poorly represented in reference databases, like proteins that are involved in very, very niche metabolic processes like hydrazine synthase. So this is why you need to be careful with your annotation. And annotating an entire metagenome is a lot more computationally expensive than annotating a single genome. So it's why if you have a specific interest in one genome in your metagenome, it's a better idea to take it all the way through binning and then refine that bin as much as you can manually, and then roughly annotate it quickly, but then annotate it properly using BLAST or Tremble or something like that, where you can really go through each sequence one by one. Okay, so you can use this output from CheckM for these kind of results that we talked about at the very beginning, right? Taxonomic profiling, comprehensive annotation, scaffolding, or reconstructing metabolic pathways, or comparative genomic analysis. So this is kind of the output of the whole process that people are usually hoping for when they do a project. Okay, so for metagenome assembled genomes, then this is the goal of a lot of projects these days. It's the goal of a lot of meta-analyses, and it's the goal of a lot of environmental sampling projects that are happening right now, like in oceans and the Arctic and stuff like this. So there's a lot of things. If this is your goal, is to get a better metagenome assembled genome from your environment, there's things you can do to improve your metagenomes, your metagenome assembled genomes, and that is to go back to the unbid contigs and the outliers, because a lot of times you can lose a contig that's like 5 or 10 kb, and that contig turns out to be the bridge between your two much larger contigs. Another thing you can do is look very carefully for the SSU in the bins, because sometimes an SSU will get misbinned, and that throws off when you try to reassemble the entire bin. So it's a good idea to use a few different tools, like we use SSU Finder, Barnap, and RNAmer when we have a set of bins that we're very interested in, just to make sure that we're not missing anything, because they each have their different blind spots. And then you can pay attention to bins that are highly incomplete, like the ones we saw earlier, where there's 3 different bins that could potentially have been the same genome, but you have to really take some time to look at why those contigs were separated. And there are several tools that are available that you can help to do this, like Busco, we've talked about a few times already. Unicycler and Tricycler are very popular, but depending on the heterogeneity of your metagenome, they have different rates of success. So Unicycler in particular is not great for a highly enriched metagenome. It's basically just like running spades. It can improve it a bit, but not always. Circulator, I've not always had a lot of luck with, just because I have a little bit of trouble getting it to run. But Jorg, I actually highly recommend, but it's not a good tool to use for anything other than a single genome, because it uses NERA, which is an OLC assembler, and you can't run it. If you have too many genomes in there, it'll just crash any kind of computer, because it just the memory use doesn't scale. So it's good for a single genome, but if you're trying to do more than that, it's not a good idea. Okay, but there really is a way that you can get much better metagenome assembled genomes, and that's right back to the very first question that we asked when you're talking about your project, and that's to think about your goal. So if your goal is to get an assembled genome out of your metagenome, the best thing you can do is save your money and get shallower sequencing for your Illumina sequencing, because the hybrid sequencing approach really does yield superior scaffolding and the assembly process. So what your real goal is to not get a comprehensive picture of the metabolic potential of your sample, but rather to extract a closed genome from an enrichment culture, you can improve your chances of that dramatically by paying a little bit less for your Illumina sequencing and using that money for PacBio Sequel 2 or the new one. And if you get your Illumina sequencing at a coverage, a total metagenome coverage of less than 100, you can usually get it quite a lot cheaper. And then you can get your PacBio sequencing on top of it, and you will get way better genomes. Okay, so that's my talk for today. Yeah, so I can see a few questions, but again, I'm sorry, I can't read them. This is not my laptop, so I don't know how to find the translate tool. So maybe if Prof or I saw KK in here can translate for me, or somebody else can ask in English.
The chat is not important.
Oh, okay.
It's just I think somebody should not belong to this group came in. Just advertisement. So.
Oh, I see.
Yeah. So.
Okay.
Any question? 有问题吗？
有什么问题可以提问？
这个讲的东西其实还有一点难理解，因为毕竟是讲那个你用 high speed sequencing 怎么去做这个 data 的分析。其实他就告诉你说其实我们 try 了不同的那种 pipelines form， 然后你就发现呢，你用这个 DAS and also 那个 check M 这两个就是非常好用的，而且最后这个 conclusion slides 也非常重要。它重要那种 hybrid sequencing， 就是说你要花很多钱做一个 deep sequencing， 它到非常 high 的 coverage， 比如说coverage 大100或者是500，比如说它会让你的 coverage 超过500次，就是来回 sequence 超过500次那样子，每个 sequence 那个就很贵了。它的其实我们就是经过很多的那个分析呢，就发现你不用就做 deep sequencing， 尤其 for the shotgun meta-genome sequencing， 然后你就是比如说少于100倍的 coverage 也是可以的就是你可以比如说2个 reset 2个公司，对吧？因为它有不同公司有不同的自己的优点嘛，然后把结果放在一起，可能会更准确。这是他整个的那个讲到后的一个结论。你们有问题吗？这有问题，你要是对中文可以问，现在也可以，或者说等一下也可以。
我有问题。
有问题吗？
我问一个。
你问一个，那你问一个，你问 Matt 好了。
Hi Matt。
你要学着老师的。
Hi Matt, are you there?
Hi. Yes.
Yeah, I'm Dan, and I'm the host of today's lecture. I have a question for you. That in the last part of your talk, you mentioned that you use the metagenome from the highly enriched animal culture, but you didn't get this slice, but you didn't find the pathway of the animal's functional gene.
Yeah,
because it's not marked in red. So can you explain it?
Yeah, so that's because this annotation pipeline is a dirty one. So we use AGNOT mapper, and the AGNOT mapper database is maybe a little bit old, right? So it might not include this sequence in it, or the sequence might not match it at a high enough identity for, because this is not the complete analysis that we've done for this project. This is just a demonstration for here. But when we take this set of bins and we spend a little bit more effort. So for this project, when we're doing the actual project, after taking the bins from check M, then we will manually curate the bins by going through them one by one to look at the RNAs that are in the bins, and to find any bins in the unbinned bin that might contain sequences that could belong to Quininia. And then we use Jorg. So Jorg uses Mira to do an OLC assembly. Basically, Jorg calls Mira bait. I don't know how familiar you guys are with Mira. Mira is an OLC assembler that was developed by the Sanger Institute, like in the early 2000s. It's a very old assembler, but it's one of the few remaining OLC assemblers that can really handle this level of sequencing that we have now, the depth of sequencing that we have now. And so it uses Mira bait to match reads from the original cleaned read set. So it maps reads from the original read set to the set of contigs that you can input to Mira as like trusted contigs or something like this, and then it will scaffold onto those contigs by iterating the OLC assembler over that subset of the reads. And so Jorg will iterate this 15 or 20 times, and it can really reduce the number of gaps in the scaffolds. And then when you can annotate those scaffolds much more carefully than you can annotate highly fragmented genome, because a lot of the servers, like the servers that maintain up-to-date databases that will include sequences like these, they won't accept highly fragmented genomes for submission. So they'll usually only accept some maximum number of contigs that comprise some maximum length of sequence. So the only other way to really do it is to call all the genes in your bin and then try to blast them one by one. But again, blast, depending if you have your own database locally, then you can do it as much as you like. But if you're accessing NCBI through the API, at least at NUS, we have pretty strict limitations about how many queries we can submit to BLAST, and it's happened several times that the whole university has been blacklisted from GenBank for like a few days, just because somebody from computer science gets past the API restriction and then NCBI just shuts us off. So this kind of result is not uncommon. So this is why what you're really interested in is specific genes, like in this case, we weren't really looking for the HDS gene in this metagenome because we had already found it by PCR. So we knew that it was in there. And when you properly annotate the bins that correspond to your genome of interest, you can find these things.
Okay, thank you.
Mm-hmm.
这么多年的经验，还是 sequencing。如果你真的不会分析这个数据啊，我觉得还是就慎重去做的，因为真的是你只要是从那个公司里面那个数据啊，你不要看就是我们自己分析不同的那个有不同的那个结果会出来，然后我们知道呢，你问我们这怎么有什么，所以我们自己判断说，哎，对，对。对吧？然后我们其他的方式也从来也验证过，对，这个其实更基于它真实的情况，因为它这个毕竟是非常高通的一个数据，当你搜索的地方看的话，这个结果可以是非常不一样的。所以说这一点就是大家做那个投资办法怎么来是一定要非常。哎，
还有一个线上还有林昌，
什么问题？
Hi, Chang. I think.
Hey,
you can ask.
Kanda, I want to ask a question. Hello, Matt. Hello, Dr. He.
经常release you 啊。
Yeah, it's me. I got your the advertisement from Da that you will give the presentation. I was surprised to see Matt also here. So I was really happy to hear also Matt speaking after many years.
Hi, Ding-Chang.
Hey. So I can take my camera. So I am I was working with Dr. He and Matt. So just to let everyone else know. So I have a question regarding this Animox community analysis, where you use the phylogenetic markers to identify how many percentage are Animox and how many percentage are the others. So with these two different software, you got totally different results. How could you? What is the reason why? From the same data set, you have the estimate totally different things. And do you know the algorithms behind these two tools?
So it's really not uncommon at all. You see this with every single data set. It's because of the approach that they use. So the Kraken uses simple k-mer alignment. It uses a greedy k-mer alignment algorithm to match the k-mers to its database, and whatever is the first hit, that's the hit that it returns. And so when you run a Kraken bracket analysis on the reads, you end up with 0.000000001% of some very, very odd taxa, because it doesn't make any assembly of the reads. It just so we'll take the entire set of reads and shatter them into k-mers and then just start aligning those k-mers. And so the result is usually a very, very heterogeneous metagenome that isn't necessarily representative of the input. So you kind of have to just ignore things that are less than 0.5 or 0.3% or something like this. Whereas for the metaflan, the metaflan tries to merge the reads first, and then after it merges the reads, it matches them to a marker gene database that's a kind of bespoke marker gene database. It's different from the BUSCO database. It doesn't count single genes. It just counts families of genes in its database that correspond to different lineages. So the metaflan output builds kind of from the top down, and the Kraken builds kind of from the bottom up. So metaflan, when you get the metaflan result, it will tell you in descending levels of hierarchical taxonomy what fraction of the reads hit markers that were in that level. And so then you can pick at the genus or species level or family or class level or whatever. So it's really, because of these two different approaches, the k-mer matching versus the marker gene database matching, you end up with this type of result. And then for BUSCO, like for CheckM, because BUSCO uses single gene copies as an evaluation of genome completeness, depending on the composition, like how compositionally similar your genome is to the reference genome at that level within the BUSCO database, your genome might or might not actually be marked by BUSCO as belonging to that, because if it's missing too many, the BUSCO like auto taxonomy assignment, which is what CheckM instantiates, will call it as the level up above. So that's why, like for some of them, it will just call it as like bacteria, right? So you have this one here that the SSU that's in the bins get matched to the Planktomycetes, but the BUSCO lineage just calls it bacteria because this Planktomycetes doesn't match very well to any of the other marker lineages in the BUSCO.
Yeah. Okay. Thank you. So it means that there's a lot of randomness in this type of analysis, right? Would you then trust more the results that you showed on the table above? So I mean, it also kind of showed the community, right? This is after the assembly and the binning, then the quick tools that are listed below.
It's difficult
the
to
one
say.
above is more computational intensive.
I think it's risky to say one that you trust more. That's why for us we run so many different. The way to actually build confidence, if what you're interested in is the phylogenetic composition, is to match marker gene sequencing with the shotgun sequencing, because that can really help to guide the annotation as well. Because if you can get a much higher resolution of taxonomy from marker genes, because that's what it's for, right? So here, if you're really curious to find out which of these is so-called correct, then you would need to either do marker gene sequencing, or you could do qPCR and try to find relative abundance if you've got a target that you can use for whatever lineage you're interested in. But I wouldn't say that one is right and one is wrong. That's the problem with these analyses.
Okay, thank you. So really nice talking to you. And
yeah, nice to see you, Ding Chang.
Ding Chang, next time maybe you can give a talk here.
I already will give a talk.
You met him already?
Yeah.
Oh, no, through me. 啊，这个就是一个非常，也是挺让你去感受很多很多一个东西。但是你真正的可以这些数据的话，他们可以是个不管让你很糊涂，但是还知道怎么弄。时间到了，那就如果有问题大家可以。